Hello, welcome back to another science stream. Magandang gabi po, madlang people. Ako nga pala si Beanie Stacy. Aya, Mika, I'm kidding. This is Millennium. Hi, I'm Millennium Isekai'd Mask Knight Empress Scientist, and today we are going to discuss the cell cycle. Okay, cell cycle. So let's go. Marker time. Okay, cell cycle. So, this one has a lot of steps in a circle. So, let's start. So, the first one would be your interface, right? Yes, hello, Decipher. Ohayo gozaimasu. Yes, we are starting with the cell cycle today. For the jumping jacks, aka set 8, it'll be about after 30 minutes. Just so you don't reverse where your food goes, because I just recently ate and I was like, maybe it's not a good idea to jump around. Like, okay, I'll save that for later. Oh, what is that crown thing? Do you see it on your end? Is it crown number 1? Is that like a... Oh, it might be on the desktop version. Yeah, okay. Yeah, so for your interface, this one is G1, S, and G2. G1 is the cell grows in size. Oh, how's the mic, by the way? I think it works, right? It works. Grows in size. The organelles are synthesized. And the cell just does its normal function. Cells do their own function. Because it's like this. Interface, mitosis. It is working. Thank you for confirming. But just in case, because sometimes I might accidentally click the mute button. Is not a button. It's like an area of the mic that's just touch screen, I think. Yeah, so it's a weird one. So for mitosis, that one has PP mat, and then there is cytokinesis. So again, cell cycle, G1, S, G2, mitosis will be PP mat C, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Organelles grow in size, synthesis. Oh, before that, you have your... Oh, I'll just write the checkpoints later. S phase is synthesis. This is where what's this called? This is where DNA replication occurs. DNA replication. Histones and other stuff would be produced here. And then for G2, that's actually where the cell grows more. All right. There are certain gates. You have G1/S. You have G2/M. Gates, aka checkpoints that they need to go through. G1/S just checks if your cell is ready to replicate. And if it's ready, it will move on to your synthesis. G2/M checkpoint would be if your cell is ready to divide. That's what happens in mitosis. So regarding the specific stuff in mitosis, again, I said PP mat C. Well, specifically just PP mat. This one is prophase, prometaphase, metaphase, anaphase, and telophase. Okay. So for prophase, this is where your... What's that called? I know that this is where it would condense. Okay, how it looks like would be your DNA would condense into very, very visible chromosomes. Like that. They become visible under the microscope. So, visible chromosomes. And then your envelope, the nuclear envelope, aka where your DNA lives, aka the nucleus, would slowly break down, but in the pro-metaphase, nuclear envelope has fully broken down. So it's gonna be gone. Now, oh, and the chromosome now attaches to the spindle. Like this. You have some stuff there. But note, it is in the metaphase where they would all line up in the center. Okay? And then anaphase is where they break up, they split up. Telophase is when, what's that part? The nuclear envelope will start again to reform like that. Cytokinesis is when it would really separate already, and your nuclear envelope is now reformed as well. So that is the cell cycle in order. And as for the time, your G1 would be 11 hours. I'm gonna write it in a different color. In blue. Why not blue? 11 hours. 11 hours. S8. G2 4. Mitosis 1. So is that 24, 11, 8, 19, 20, 21, 22, 23, 24. Yup, there you go. That's 24 hours. So again, G1 11, S about 8 hours, G2 about 4 hours, mitosis about an hour. For what species? Humans, obviously. Humans. So we are going to write it again, just to make sure that it is installed in the brain. Okay. Okay. One more time. Let's go. G1, G2, G3. One more time. Alright, what were we discussing? Cell cycle. Cell cycle. What's the first part? Interphase. With what's here? The G1, S, G2. Mitosis would be P, mat, C. And then you have, oh, no. I already wrote it. And cytokinesis. G1 is where the cell prepares to grow in size, and organelles are made, organelles are synthesized. That's not a word. Organelles are synthesized. There you go. Organelles. S1, S is when your DNA would multiply and just condenses into your DNA replication occurs. Now it's times 2. G2 is when it will try to just grow further. For the gates, you have, oh, I forgot to write another gate from earlier. G1/S, G2/M, and you have your M/A checkpoint. Okay, I'll write that a bit. That's just to check before they split up. Like if it's really fully in the middle and all the things are attached. Yeah, that's the other gate. G1S, G2M. Around the 30-minute mark, and I will have to set an alarm for the jumps. G2 is just grow further. Prophase is when the nuclear envelope starts to break down. Prometaphase, it's fully breaking down, fully broken down, and they will attach to the spindle. Okay, metaphase, it's in the center already. Then the checkpoint will check. Anaphase, they split up. Telophase is when the nuclear envelope would start to reform. Telophase is starting to split up. plus the actual cell itself. Cytokinesis is when it's like really, really splitting up. Okay, now specific details. What is Rho AGTPase regulates contractile ring formation. What's that about? It's about cytokinesis. Rho AGTPase. Now for this one, I just want to highlight again how long this takes in a human. It's gonna be 11 hours for G1, 8 hours for S, 4 hours for G2, and about an hour for the rest of these. So 24 hours for humans. Yeast is about 90 minutes. Yeast. 90 minutes for yeast. And then embryo is actually about 30 minutes. And then E. coli, 20 minutes. Okay, so 24 hours human, 90 minutes yeast, 30-minute embryo, 20-minute E. coli. One more time. 24 hours human, 90-minute yeast. Yes, very good. Those emotes are really nice. The notebook and the eye thingy. 24 hours human, 90 minutes yeast, 30 minutes, and 20 minutes. 30 minutes. What was it? Embryo. 20 minutes E. coli. Good. Now, what are cyclins? Oh my gosh, where is my sticky notes? Okay, there you go. So this is the in the head, of course. Thank you. Yes, notes, take notes. These are incredibly important. Very. Why did the heart rate reach 121 when I didn't even do the jumping jacks yet? It'll happen. Soon it will happen. I just need to make sure that the chorizo is properly digested first, about 30 minutes to an hour. We do not want our appendix to explode, right? Okay. Now, picture. Wait. Okay. Now, what are the cyclins and how does it fluctuate? Wait, what is my hat right now? I want to change that. I do not want the water bucket. It has to be the dab. I want the dab hat. Oh, not the water bucket. That's like a Wi-Fi. So yeah, we shall change the hat. So next are... Oh, that is so cute. It became the wave. Okay, that's nice. Wave. Next is cyclins. What are Whoa, wrong color. Now, what are cyclins? Cyclins and its fluctuation. Well, the marker is so thin and it's not clear. Cyclin. Oh, that's much better. Okay. Cyclins. And how does it fluctuate? So, you have the activation of cyclins. The activation of cyclins bind to and activate your cyclin-dependent kinases, the CDKs, via phosphorylation. What does that mean? Well, you have the CDK being inactive, and then the cyclin would binds to it, which exposes its active site. So imagine if this was the CDK is a tsundere, but if the cyclin, the main character, attaches to the CDK, look at this. If the main character / the cyclin would attach to the CDK, then the CDK is like, will reveal its true form and it can now have an active site. And now you have the cyclin activating kinase phosphorylates a residue on your CDK. So now the CAK, the cap, can now add phosphates on the exposed active site. So what are the specific types of cyclins? It would be, one moment. Well, we can write it. The specific types of cyclins are, here you go. I can actually just, one more. I'll write that again. CDK inactive state. Can I actually just do the backspace? There you go. Beautiful. So now different cyclins. What are they? They are the G/S, S, and M. These accumulate and degrade. At different points. So you have the GS cyclin, the S cyclin, the M cyclin. But they look incredibly familiar because these are almost, look, it's like the G/G2/S, the synthesis phase, and the M phase. So when do they accumulate? It accumulates at certain points, right? So let me just draw it out here. Wait, one moment. We need a lot of space, so I'm gonna erase all of these. There you go. Erase time. The fluctuations will be drawn like this. So you have the G1, you have the S, G2, and M, and then back to what? This is a cycle. Back to G1 a little bit. This is the start of the next one. So I mentioned earlier we have 3 types of cyclins, G/S, S, and M. We will draw them in specific colors. Blue being who? G/S. It will be a little bit high here. And then boom, low. Kind of like this. Wait, let me write that again. So it kind of increases here and decreases here. That's the G1s, the G/S cyclin. And then you have the red one, the S cyclin. Guess what? It increases here until roughly middle-ish of M. It's gonna be same here. Like that. That's your S cyclin. And then you have the M cyclin. Where, I wonder, will it be so high? It actually starts to go high a little bit halfway here. Reaches peak around the M. And then halfway M goes down. This is the same amount there. So one more time. Wait, M cyclin. So again, for the cyclins, you erase that. For the M cyclins, you have G1. Whoops. Disgusting line. G1, S, G2, M, and then G1, right? Now, this G/S will increase, kinda peak here, and then lower here. G1/S. And then M would be not the green, it's gonna be the red. M cyclin is gonna be up here. It's gonna go high here. And then go low here. Oh, this is S cyclin actually. S. S cyclin, S cyclin. And then M cyclin would go up a little bit here and then go down here. Is that right? Not so much. It's gonna go a little bit higher around here. Like that. Yeah, much better. This is your M cyclin. It's before mitosis and peaks. And then the rise. Where is it? The rise but degraded after metaphase. Yeah, because it's just 4. Oh, thank you. Are the lines becoming straighter and less cronkier? Because we're trying to make that happen. This is an art stream. This is Van Gogh level art. So we're trying to make it as pretty as possible also. But yeah, those are the cyclins. You have... Okay, there's a couple more, like the G1 CDK. Let's see, where is it? You have the G1 CDK. This one. And then it goes to the G1/S CDK, SCDK, G1/S checkpoint. The checkpoint will be a square. And then you have the... What was it? G2/M checkpoint. And metaphase/anaphase checkpoint. This is just... Remember, what's the G1/S checkpoint? It's checking if it's ready to ×2×2. See if it's ready to Kage Bunshin Jutsu. G2/M if it's ready to split apart. If it's ready to proceed to the mitosis and if it properly synthesized itself. Now, the metaphase will be... The M/A would be if it's really ready to just split itself apart. Because that is what happens in the anaphase. Now, there is a very specific thing in the M/A one that I want to highlight, and that is the APC, aka that's a very specific one because that's the MCDK inactivation complex, anaphase promoting complex. Actually, anaphase promoting complex. Oh my gosh, is this the same one in the wind? Maybe. Is it? Google. Hello, Google. APC is that the same thing? APC wind pathway. Wind beta catenin. Oh, it's a different one. This one's a protein. But the wind one is adenomatous polyposis coli. Okay, it's not this one. This APC here is the anaphase promoting complex, and the one in the wind is a different APC. But they look the same because it's the same letters, sadly. Now for the APC, anaphase promoting complex, it will what? It will promote the anaphase. It's a complex that promotes the anaphase. Voila. M cyclin marks M to A transition, and if you suppress your M CDK, it prevents premature mitotic re-entry. Anyway, these are just very, very regulated so that you don't just do the cell cycle like so much, even if you don't need it. Okay, now moving to the meiosis and mitosis section. Meiosis, mitosis. Marker for meiosis. Meiosis, mitosis. Imagine if it suddenly becomes a karaoke stream. No, hold back. Not yet. Generation. Meiosis would be the generation of haploid Okay, so let's simplify it. Right now, mitosis would be you have a circle, you have another circle, you have this one, the alleles. They would multiply, replication, DNA replication, right? And so they now become two, connected at the kinetical. And then it will split up. It will break up. At the center. And like that. Grab it. Spindle. Grab it like that. And then they become like this. Looks weird, but don't mind. And then they will split. Okay, we need more space, bro. Into two circles, which are diploids. Like this. Wow, no. Ugly. I will redraw it. It's too ugly. Neurosama would be like, no, that's ugly. Vidal, ban the mods. Or whatever. Something like that. No, mitosis, circle. Circle. If it was a notebook, the correction tape would be used up. It's gonna be ubos na after drawing this one. Okay, so you have this one. DNA replication. There you go. Good. Now they split up. Slowly. No. Go to the center first. Expand a tad bit. This is the metaphase now. Go like that. And then they split apart. They're dragging one line each. That's the anaphase, bro. And then they become two circles. That's a circle. With the nucleus having two lines. Beautiful. Now the mitosis. You have a lot more circles here. So let's start with this circle. This is generation of haploid, but I'll move the letters. Here, generation of haploids. Generation of haploid. Circle. We start with, okay, I'm gonna make this red and blue. And then U times 2. So now this red is dalawa. 2. And then blue would be 2 as well. And then they now cross over. So that is a very specific thing that happens in meiosis. Those two would cross over. / pairing. They will talk here like this. Whoops, wrong color. Like this. Let's say here, and the red one will I said red, not blue. Like that. Interact. After they cross over, right? They will now like attach this way. Yes, exactly. This mitosis is really bacteria multiplying. This is it. It is. So that's why we're drawing this one. And meiosis would be, for example, like haploid, like sperms or something like that. That would be here. And then let me just draw it properly. Yes. Dab. Let me dab too. Where's my dab button? Dab. There, that is the dab. Okay, so for crossover, they will interact because you don't want. For imagine if you look exactly like your sibling, that would be mitosis. Hello, Apu. Welcome back to the stream. Whoop whoop. Yeah, dab dab dab. We will use the emotes to the fullest. Regarding this one, if crossover doesn't happen, then you will be a perfect copy of your sibling. That's why crossover is good. So there is genetic distribution, a tad bit of a difference, and all of that. So the way that happens is now after they cross over, they will now like, and your envelope also gets destroyed. But here you get, let me see. I'll just get one here. You don't need to split it up. Circle, circle. There, one. It'll just get this red, that's not red. Yes, this is circle, but this red, look at this. Focus, focus. It has a blue. And then this blue, look, focus. This one would have a red part. Yeah! Now they are attached to the puller, the spindles, and then once they, for example, once they split up later on, this red, now one red, this red, and a red a little bit here. Yes, focus. Focus, 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 focus. No, that blue. So, we're not yet done. This is crossover and segregation of chromosomes. Segregation of chromosomes. Now, this will actually split up again. And the final thing is, we have 4. So the steps over here is now this one. Voila. Look at that. Look at that. Look at that. It's now mitosis. Look at that. Right? And the final cells here. Red. Half a red. And this one has a little bit of red. Now you're half a blue, and a bit of blue. So see, that's how those are all different. Booyah. So you have 4 haploid daughter cells. 4 haploid daughter cells. Oh, 4 haploid daughter cells. And then this one is 2 diploid daughter cells. Daughter cells just mean 2 or offsprings. Not necessarily like kids or something. It's just the term. Just the term. And then that's n equals 23. N equals 23. So here you have 46 total. Alright? Focus. Focus. So that's mitosis and meiosis. Best friend. It's a good... Okay, now. Cell cycle again. What is cell cycle? It is a process where you aim to divide into identical daughter cells. So let's just do a mini circle. They grow, they combine, they share apart, and they split and split again. Yes, it's like watching anime and the different arcs. They grow, they split again, and then they come back, and then they split again. It's like Naruto, and then they grew as a team, as Team 7, and then Sasuke got brainwashed by Orochimaru, and then he left, and then in the Shippuden he came back, but he came back as an angsty teen, and then after, if you skip to the end of Shippuden, they're back together. That's the whole show. It's very nice. I highly suggest watching it. And meanwhile, regarding growing and splitting again, you have they look like smiley faces in my notes, but these, let's say you start here. You have a daughter cell. Normal cell here. You have a cell with this one, and then you go G1 with cell growth and S multiplying. Then you have this one now here, 2. And then, no, that does not look like it's a very clear DNA. I'm gonna write that again. Like this. Yeah, that's good. So this one, and then the chromosome segregation. Like, let me see. Oh, that's a long arrow. I'm skipping a lot of steps. Just know it's like that. And then they split up into 2 squigglies. See, look. This is where the mitosis part and the anaphase part specifically happens. So that's the general picture of the cell cycle. So what's the achievements? You have the growth, the duplication, the splitting, the very, very splitting. Now we have, how do you control? Speaking of anime, do you like genre of adventure and psych? I have a recommendation. It's called Girls Last Tour. They have an anime now, but I highly suggest to read the manga. So, good question. My favorite ones are rom-com. Like Kimi ni Todoke. What was it? No Game No Life is not rom-com, but it's really nice also. And regarding adventure and psychology, like Classroom of the Elite, I finished season 1 and 2. I haven't watched season 3. I did a lot of Naruto and what was it? A lot of the Shippuden. I read the manga for the rest of it. I do not like Boruto. Not so much of a fan, but it's like I'm okay that it's continuing. I just prefer the old characters. No Game No Life has, oh my goodness, really good question. Season 1, I call it season 1 because I'm still hoping for season 2. Season 1 and the movie, they did that. No next season yet. I finished all of Sword Art Online, the anime. I didn't do the light novels, but I did all of the anime and the movies. Really, really great. So that's like, if you literally see why I have 2 swords behind me, that's a very big clue that I was a big fan. I am a big fan of Sao. And what else? Shield Hero, I watched a bit. I gave up on the turtle part of season 2 because I was like, what's happening here? Actually, I am planning one of in a future one, hopefully, where I will just yap about my anime listing and just discuss how I gave a lot of 10s or 9s or 8s for the lists. But yeah, I'm a manga reader and anime watcher. Mostly rom-com and isekai. I avoid the scary ones, the horror ones, but I did watch Jujutsu Kaisen and Attack on Titan. I was heartbroken at the ending of Attack on Titan because I was like, what's happening? Why did I waste my time watching this? It was then just gonna be painful. Jujutsu Kaisen, of course, you can't let go of the fact that Gojo is there. But the rest, I try to avoid scary stuff because they are scary. But Gojo was an exception. And Levi, especially before the last season happened, before the art changed. So that is the mini sidetrack of it. Yeah, going back to the 2-step control. Wait, what was the girls' last tour? Is that a scary one? Because if it's scary, I cannot. But yes, that is the general picture of it. Yep. Okay, now we will do the 2-step control of replication. / initiation. Oh, I have to write that down. Well, no, it ain't scary. I will not spoil though. Thank you very much. Yeah, I will. One moment. I will list it down for a future uh, drawing or yapping stream just to discuss my anime list. And that includes manga as well. Because that's gonna be a very fun one. I don't know if it's gonna be a fun one, but I will talk about some anime. I'm like, okay, either way, well, it's gonna be out there. So two-step control. What is this? How to ensure that you have a single round of DNA replication. So G1, and S, and M. Where's G2? G1. This is supposed to be G2. Oh, this is G2. SM, G1. It's just cycle 1 and then cycle 2 at the bottom. Licensing. So, licensing. Pre-replicative complexes are assembled at the replication origins. Helicases are loaded onto DNA during this phase. Helicase. What do? They split things up. They split apart the DNA. S would be cyclin-dependent kinases. Helicases are activated and form the forks. So here, helicase loading. Helicases are activated and form the forks. Here's the thing. Phosphorylation prevents reinitiation at the same origin until at least the next cycle. And then cohesins hold the sister chromatids together. So that would just be, let's say there's the DNA, and then you have cohesins like that going around it. Okay, and then you split them up. Now you split them up, like that, split into line, line, and now you start. Note, condensins compact the DNA of the sister chromatids. They just have different functions, but just remember, helicase loading is the licensing, and then helicases are activated after. Centrosomes act as spindle poles. Okay, and organizers during mitosis. Good to know. Good to know. Good to draw. Draw it. Or no. Helicases. So you have centrosomes act as spindle poles. You have kinetochores, right? Kinetochore protein complexes on the chromosomes where the microtubules attach. Let's say this is your chromosome. You have the kinetochores here. That's where the microtubules will attach and they can split up. Motor proteins would facilitate microtubule splitting, aka they are the ones that will pull stuff like that. Help the pulling. Motor proteins. For the motor proteins, you have kinesin and dynein. What's the difference? Dynein is retrograde. Kinesin is anterograde. Right? Dynein. What is this? It could be pull or push, but because it depends on the type. But just remember, dynein is retrograde. Kinesin is anterograde. DRKA. And then each kinetochore can capture multiple microtubules at their end. And then anaphase, for example, you have at anaphase A, you have the sister chromatids being more toward the spindle poles due to the kinetochore. Now, for the I drew this earlier. We mentioned how the CDK is tsundere. So, This is a CDK, your normal tsundere. And then if a cyclin attaches to it the main character. Hello, Sheriff. Welcome back to the stream. We are talking about the mitosis, cyclins, and all of that. Side note about the jumping jacks, it might happen really soon. I didn't do it at the beginning of the stream today because I needed to digest some food and I did not want to reverse the order of natural flow. Okay? And now we're talking about how the CDK is actually a tsundere, and if the main character cyclin is bounded to it, it will activate the CDK and expose some stuff. Now you have the cyclin activating kinase. It can phosphorylate the CDK and add the phosphate to fully activate it. I'm not sure if you could see it, but can anyone see that the crown stuff happening there? I think this one's available only on the computer version of YouTube. But yeah, that one's nice. Because you could see it. It's like, whoa. You gain experience points. I don't know if there's a new feature that they just started rolling out to the different channels. Oh, side note. Let's go and discuss how mitogen signaling reactivates the cyclin CDK. How mitogen signaling reactivates CDK. Cyclin CDK. So your mitogen is a growth factor. Oh my goodness. We need space for this one. Here we have space. And it's gonna be like this. Mitogen goes here. And then your... What's this receptor? Mitogen receptor glows and talks to the RAS. Ooh. It talks to RAS. So this is similar to RTK. So it talks to RAS GDP. Then RAS GDP, RAS is activated and then talks to RAF, Mech, Irk. Okay? This is now RAS. RAS talks to RAF. RAF is the map KKK. Mech is map KK. Irk is map K. So here is just a cycle of ATP to ADP with double phosphates going on over here. ATP to ADP, double phosphates going on here. And after that, you activate the transcription of some transcription regulatory proteins, right? Because it's a kinase, so it puts a phosphate. And you go inside the nucleus. Let's say there's a gate here that's open. Goes inside. Now, the MAP kinase triggers the expression of immediate early genes. Let's say this is an immediate early gene, such as the MYC transcription factor. Okay? It's made and translated. MYC. Whoa. My goodness. MYC drives the delayed response gene regulation, aka promotes transcription of cyclin D. So MYC, again, this one is mitogen. So MYC promotes the translation, the making of cyclin D. Cyclin D complexes with G1 and CDK. Again, Rasraf McIrk makes MYC induces the transcription of your cyclin D. Cyclin D attaches G1 CDK. G1 CDK phosphorylates some stuff like EF, G1 SCDK. Essentially, Mick is really deeply involved with a lot of the G1S CDKs and SCDK, so that's why you don't want it to go bad, because your Mick is it's one of the very common things you see in cancer, because if it goes bad, then your cell cycle is just, it goes wild. Now let's talk a little bit about the tumor suppressor P53. Tumor suppressor P53. Here. Okay. So tumor suppressor P53, we've heard of it a lot of times. Okay? Why? Because P53 prevents the G1 to S phase progression, especially when DNA damage is detected. So, if DNA damage is detected, P53 should be able to stop that cycle from continuing. Okay? So, let's say you went outside without the sunscreen, or you got too many x-rays for the day, for a year actually, so you get DNA damage. Okay, now what happens after DNA damage? Initiates your ATM/ATR kinase activation, and then your ATM/ATR would phosphorylate and activate the CHK1 and CHK2. Just know the letters. And then, in normal condition, let's see. In normal condition, that is a note. Normal, P53 is degraded by MDM2. But let's see, here. P53 is normally degraded by MDM2. So P53 degradation. But if the CHK1 and CHK2 are activated, then the MDM2 is removed, and your P53 is phosphorylated and very activated, and it can bind to and make the P21 protein using the P21 gene. Transcription and translation into P21 gene. Now, if we made, P21 protein. If you made the P21 protein, what does that do? It will bind to your G1/S and SCDKs. So it will help halted cell progression at the G1S checkpoint. So again, if you have DNA damage, I'm gonna close my eyes so I can help explain this in my head. If you have DNA damage, then that DNA will lead to ATM and ATR phosphorylation, and then which leads to your CHK1 and CHK2. CHK1 and CHK2 would then lead to phosphorylation of your inactive P53, so MDM2 gets removed from it, and P53 is now free to make the P21 protein using the P21 gene, and then with the P21 protein, that can then try to halt the cell cycle, because it will lead to the binding of P21 to your G1/S. So that one is the whole cell cycle set. And I will now before I do the jumping jacks, this is a very, very short explanation of what is actually happening. So we recently reached 12,000 subscribers. Do not mind the fact that it's now 13,000. It was at the 12,000 mark that I was like, why not? I go and to celebrate, I do 1,200 jumping jacks. Okay? 1,200 jumping jacks. And I was like, it would be crazy if I just do 1,200 in a day. I don't think I can do that. So I was like, split them up. 120 per day. AKA 10 sets. So not necessarily every day, just 10 sets total. And we finished the first, second, third, fourth. Why not count in French? On the trois, quatre, cinq, and then 6, and then set. 6 and set. I finished the 7th set. We are now, so far, how many did we do? 120 times 7, 0, 14. We did 840, and now I'm gonna do the 8th set. Yeah, right now. Like at this moment. Because I think we digested the food. So yeah, I'm just gonna check if the heart rate monitor is attached. Indeed, it is attached. So let's go. Okay, is it working? I think it's working. The set stretching time. All right. Now the 8th set of jumping jacks. Whoop whoop. And then let's just move this. Okay. Let's go. 1, 2, wait. I'll brace here. Wait, 1, 2, 3, go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40. We did 40. Let me breathe. One moment. Okay. 40, 40, 40. Remember 40. We did 40. 40. 1 second. Almost there. We are a third. 1/3 now. Okay, another round 2. 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60. That's 60. Halfway. Almost there. 60. And let's go. 61, 62, 63, 64, 65, 66, 67, 68, 69, 70. 70 now. It's almost there. Why did I write that? I was like, why? But this is healthy, right? Okay, 70. And let's go. 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85. 85. Almost there. 85. 85. And how many left? 15 plus 20, 35. Let's go. 86, 87, 88, 89, 90. 91, 92, 93, 94, 95, 96, 97, 98, 99, Hundo. The last 20, I will count in. Hi. The last 20, I will count in Chinese. Because why not? Wow, it's reaching 153 maximum. Crazy. We're at Hundo. Last 20. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We're sure. 11, 12, 13, 14, 15, 16, 17, 18, 19, Archer! 120. Tapos na. Okay. We reached 120. Whoop whoop. Alright. So yeah, I think for the cell multilingual, yeah. Imagine French counting is so hard right now. The on deux trois quatre. I cannot say number 4 properly. But yeah, for cell cycle. It was cell cycle, right? I already like the jumping reformatted it. But yeah, for the cell cycle, we finished for which record? Is it the where's my thing? Which record? Is it the heart rate? The highest heart rate I think was like 163 or 167. It's in the very first set. When he said. But yeah, I think for the cell cycle, that will be all actually for today. Thank you for coming back and see you in the next one. Bye.